Now, I think we're going to skip the video for now and go right into Andrew's talk. So our next presentation, we're going to hear from Dr. Andrew Landstrom, who's a pediatric electrophysiologist and a physician scientist at Duke University. He's going to give us a research update on SADS conditions, a sneak peek into tomorrow. Welcome, Andrew. And go.
Thank you very much, Marcia. Can everybody see the slides and hear me okay?
You're good.
All right, thank you. Well, it's a pleasure to be here. Happy Saturday to everybody. These are my disclosures. So over the next about 25-30 minutes that we're going to have together, we're going to talk about what I think are some really incredible scientific advances that I think you may not see tomorrow, but you may see in the years ahead. We're going to set the stage by talking about what I think are some groundbreaking advancements that have happened in the past 5, maybe 10 years. We'll talk about a couple of examples of, I think, very exciting new research in diagnostic testing, and specifically we'll talk about a blood test for arrhythmic cardiomyopathy. We'll talk about potentially new ways to do exercise testing for CPVT. We'll talk about some potentially really exciting updates to gene testing, particularly for BRUGADA syndrome and something called polygenic risk. We'll talk a bit more about CPVT and long QT syndrome, but some new sort of artificial intelligence ways of potentially figuring out variants that are uncertain to date, which I think is exciting. And then, as Dr. Etheridge just mentioned, we'll talk about some new methods to treat these diseases. Well, you've heard a little bit about sort of long QT gene therapy. We'll talk about BRUGADA syndrome gene therapy potentially, and some new models for testing new medications in arrhythmic cardiomyopathy. So I'm excited to be here, and let's get started. So as both a physician and a scientist, sometimes I feel like those two sides of me are at odds, because what I really want are scientific breakthroughs to help patients that I see. But those scientific breakthroughs take a long time. And I sort of am kind of an impatient person. I want them now. I want them kind of right at your fingertips very early on. But it's important to remember that the science can be slow and a long road, just like this. And the reason being is that we have to be able to get it right. So a scientific breakthrough in the lab doesn't necessarily translate to something that's really, really good for patients. That process takes a lot of time. And so everything I'm going to show you today are breakthroughs in the lab or breakthroughs on the clinical side, but not necessarily something that's reached the endpoint, the destination of being ready for treating families with SADS conditions. So 2022 is an exciting time for science. And in my opinion, there are 4 big advances that have happened in the past 5 years or so that have really set the stage for all of the really cool advances that we're going to talk about. Let's go through kind of what those big breakthroughs are. One of them is how easy and how fast and how cheap it's become to sequence the human genome. So this is a graph. The y-axis here is dollars, and the x-axis here is time. And as time has gone on, the cost it takes to sequence a human genome has gone from $100 million back in 2001 to today about $800. At least that's what it is at Duke. And that is a precipitous fall. And that has really opened the door to ask new questions about genes, which I think are driving some of the breakthroughs that we'll talk about today. The second major advancement that's happened is something you may have heard about called CRISPR. What CRISPR is it's a pair of molecular scissors that can cut DNA, including human DNA, in a very precise and targeted way. And if done in the right conditions, can replace that DNA that's been broken with something else. You can swap out pieces of DNA, you can add pieces of DNA. And this has implications for how we study genetic disease, but also potentially how we might treat genetic disease down the road. The third major advancement, and you've heard a little bit about this from Dr. Etheridge, is gene therapy. Now, gene therapy is something we've been talking about for many, many years, but I think it's really getting to the point now where the science is advancing fast enough that we're at a threshold moment where you're going to start to see this more and more in the clinic. And we've got some really cool examples of patients without SADS diseases who have benefited from this, but I think you're going to see this in the next 5 to 10 years, as Dr. Etheridge is mentioning, really sort of creep into the SADS space. And I think this is a very exciting potential new field of therapy. And then finally, the ability to learn about SADS disorders has been greatly enhanced by this discovery. These are beating cardiac cells, beating heart cells derived from a patient. And rather than trying to take heart pieces from people, that's really not something we can do. This technology is something that's revolutionized our ability to study cardiac disease. And with a few drops of blood, you can isolate a particular kind of blood cell that grows in the lab and then trick those blood cells into becoming stem cells. These are called induced pluripotent stem cells. These aren't embryonic stem cells. They can be made directly from blood cells. And those stem cells want to become every cell in the body, and we can guide them into becoming cells like this, heart cells. And this has revolutionized our ability to study SADS disorders. So these are the 4 breakthroughs, and I think you're going to see echoes of all of these in our case examples today. So let's talk about what are some exciting new diagnostic testing tools that might be coming down the pipe. Well, some of you may have arrhythmic cardiomyopathy, which is defined by the fat in the heart that's replacing heart muscle. And this fat in the heart causes 2 things. It can cause the heart to not be able to squeeze well, heart failure, and it can cause arrhythmias. These arrhythmias can be life-threatening. And as Dr. Etheridge was mentioning, we don't have really good tools for fixing this disease. We'll talk a bit more about that. But one really interesting possible tool that's coming down the pike is, can we detect this early with a blood test? So right now, our best test for figuring out who's at risk of arrhythmic cardiomyopathy is the genetic test. You carry the gene mutation. But what about families who have arrhythmic cardiomyopathy, but they don't have a gene mutation that can be identified? How do we know what children may or may not be at risk in that family or siblings of those individuals? Having a blood test would be really, really helpful. And this is work by Bob Hamilton and his group up at Sick Kids up in Canada. And this was his paper from 2018. This was a really exciting paper. What you're seeing here are Western blots that basically look for a specific protein. And every time you see a black line here, that's that specific protein. This specific protein is one that has been linked with the development of arrhythmic cardiomyopathy. And so if you've got definite arrhythmic cardiomyopathy, the presence of all these black lines indicates the presence of this biomarker. And that biomarker is largely absent in individuals who don't have any evidence of disease. So this is evidence that perhaps there's this biomarker floating in the blood, which can be detected as an early warning sign for arrhythmic cardiomyopathy. And the really cool thing is that it can be detected in individuals with borderline arrhythmic cardiomyopathy. So ones that don't quite meet criteria, diagnostic criteria, this still can detect that they're at risk. And so rather than undergoing surveillance cardiac MRIs every year or echocardiograms every year, could we augment this with a blood test to look for this protein down the road? And that's really exciting for me. The other possibility here is, Bob Hamilton hasn't stopped with just arrhythmic cardiomyopathy. He's starting to ask questions about things like BRUGADA. Could other SADS disorders be detected through a blood test? And so we'll have to wait and see what his group does in regards to this, but some really interesting data that perhaps there might be a blood test for arrhythmic cardiomyopathy. Well, what about CPVT? So as you all know, CPVT is because a particular ion channel inside the cell called the ryanodine receptor leaks calcium, and mutations in this gene can cause CPVT, or mutations in proteins that change the channel can cause CPVT, and this causes calcium leak, and this leak triggers arrhythmias. Well, the trouble is sometimes patients with CPVT, they're really hard to diagnose. You put them on a treadmill and maybe they have the mutation, but they don't have any signs of arrhythmias or ectopy. And so can there be a better diagnostic test that might detect those individuals with so-called hidden CPVT? So the top panel here may seem familiar to many of you, especially those with CPVT, but you go into your provider's office and they say, okay, let's see if our medications or the sympathetic denervation surgery is working to control your CPVT, and they put you on a treadmill. And the usual treadmill is you start slow and you speed up over time, and they crank up the gradient and you run faster and faster, and all the time they're watching to see if they see evidence of arrhythmia. Well, there are many people for which this test will be negative. And it leaves the question, well, do they have CPVT that's simply hidden? And can we think of a better stress test to elicit these rhythms? And this is work done by Andrew Crane and Shu Sanatani at BC Children's, as well as other groups, have shown this. But maybe rather than starting the exercise test slow, maybe you start off with a burst. So you start off running as fast as you can right off the bat. And that burst of exercise early on has been shown, at least in these early studies, to elicit the ventricular tachycardia that can lead one to believe that you have CPVT. So perhaps in the next few years, if other centers start to adopt this and see those same results, maybe you'll see a bit of a change in the stress tests. What about advances in gene testing? Let's talk a little bit about Brugada syndrome and polygenic risk, and then the role for artificial intelligence here. So Brugada syndrome, as many of you know, is defined by a loss of the sodium channel, oftentimes with mutations that lead to the channel not being expressed as appropriately in the body. But it's, one of the things that has always kind of made Brugada syndrome really challenging is, although SCN5A is known to cause Brugada syndrome, most patients with Brugada syndrome will actually not have a mutation in SCN5A. They clearly have Brugada syndrome, yet no mutation. And it's been really, really hard to find the other single gene that's been the cause of Brugada syndrome in these individuals. That's question number one. The second question is, why are families so variable? So, I got a little bit of this from the Q&A session previously, but you may have one individual with an SCN5A mutation with really bad Brugada syndrome. They always see the Brugada pattern like this on an ECG. And other people in the family with the same mutation who never show signs. So why is that? And you could expand this question to, why is long QT different in the same family? Right? Why is arrhythmic cardiomyopathy different in the same family? Well, I think probably some of this has to be explained by the other spots in the genome that we can now assess. And this brings up this question of polygenic risk. And all that means is, we are the summation of the genetic impact of not just one misspelling in the genome, but potentially lots of other changes in the genome. We know that there's millions of differences between individuals. That's just who we are in our genome. And could those other smaller differences influence disease, such as Brugada syndrome? And the answer is yes. So this is work done by Connie Bazina and a very large coalition of investigators who brought lots of individuals with Brugada syndrome together, sequenced the genome and said, okay, what are all the genetic variants that are in some way leading to this disease? And they took nearly 3,000 individuals with Brugada. And I wonder if maybe some of you who've signed up for research studies to participate might be actually part of this study. But they found some really interesting findings. On the y-axis here is the strength of association of a given area of the genome with Brugada syndrome. And so the taller the peak, the stronger the association. And then on the x-axis here are the chromosomes, the human genome. And so across the human genome, you can see there are peaks. Now, the biggest peak is SCN5A. No surprises there. We know that gene's associated with the disease. Look at all these other genes: HA2, TBX20, GATA4. All of these genes are associated with development of Brugada. Doesn't mean that every single variant in those genes will cause Brugada, but perhaps an indication that the genome is not just one gene leading to Brugada syndrome, but perhaps lots of different genes and variants in those genes are influencing a disease phenotype. And perhaps this could explain some people who have Brugada but don't have an SCN5A variant, or variation in the family. Some individuals might have variants in some genes that make them a bit more severe than others. So this is a very exciting possibility to be able to detect this. What this allows us to do is if we can understand an individual and all of their genetic variants, and we know what the risk is of all those genetic variants for that person, we can then calculate risk and start to ask questions like, for you, how severe will your Brugada syndrome be based on your DNA? Now, nothing will ever explain Brugada syndrome completely, but this is a real possibility to be able to describe risk and give probabilities based on your entire genome, a true risk calculator, something that's really exciting and to watch out for in the future. What about long QT syndrome, and what are we doing to advance the field of gene testing here? Well, as many of you know, long QT syndrome is due to mutations in genes like KC and Q1 these mutations disrupt cardiac ion channels like here, like the potassium channel, that can lead to arrhythmias. Many patients with long QT will have mutations in these genes, but sometimes, and it's not infrequent that you actually get a genetic test report that's uncertain or unclear. This is one genetic test report with lots of unclear results. So here, the results at the top are uncertain, and it's uncertain because you've got all these genetic variants, all of which are of uncertain significance, VUSs. What do we do with these? VUSs are hard. VUSs are not clearly disease associated, otherwise they'd have been called pathogenic and labeled as such, and they're not benign enough for us to discount them. So they're things we really kind of struggle with, and you may have heard of VUSs or even have a VUS yourself. Well, I think one of the most amazing advances recently has been the ability to use things like artificial intelligence to start asking, well, maybe there's something that we're missing as humans that maybe a computer might be able to help us out with. And can we potentially eliminate these variants of uncertain significance? So this is a study that we did with Ricardo Hineo and Rachel Dralos, who are computational scientists here at Duke. And then we had a wonderful and amazing cardiology fellow, Jordan Ezekian, who led the study from our end. And they came up with an artificial intelligence platform that we call Genesis. I didn't come up with the name. I am not that creative. But Genesis is essentially AI platform called Gene-Specific Machine Learning Models for Variants of Uncertain Significance. And we took VUSs in genes that you may have heard of, KCNQ1, KCNH2, SCN5A, and RYR2, and we literally had a black box. And it's a black box because I can barely turn my computer on, and so I don't understand how they made this machine learning model. They used fancy words like epoch, multi-layer perceptron, position-specific scoring matrix. At the end of the day, they ended up with this model, and this model was actually really quite good. Nothing is perfect, but for example, it was able to predict overall, you could think of AUC as about success rate, 87% accuracy of predicting VUSs, variants of uncertain significance, as being either pathogenic or not at the end of the day. And so here at the top are those accuracy measurements that we made. So 87% for KCNQ1, 83% accurate for KCNH2, SCN5A 79, and RYR2 86% for predicting these VUSs. And if you look here down to the lower part of the screen, these are kind of the best mutation prediction models to date, and we were able to kind of improve upon that. Now, nothing is perfect, and I think this model has a long ways away from being something that is applied to gene testing for patients directly, but I think evidence that we're moving in the right direction, that we can utilize things like artificial intelligence potentially in a diagnostic way for genetics. Well, what about new therapies? And Dr. Etheridge has spoken to you a little bit about this. I'll try to expand on a couple of different things because I think this is perhaps where some of the most exciting advances are going. So if we go back to arrhythmic cardiomyopathy, we talked a little bit about this previously, that we really can't do anything to slow the progression or reverse the disease of arrhythmic cardiomyopathy. And it's sort of the case for all of the SADS disorders. And this is really a big problem. And I think one of the reasons why there are no specific treatments for AC is that we really have a hard time with models. There are some cell models and there are some mouse models, but sometimes it's really hard to translate those findings that work for a mouse into humans. And so I think the key to figuring out new diagnoses is how can we actually come up with new models that are informative and maybe are patient specific, so that when we learn something about a patient, we might be in a better place to generalize it to others. And so I think new models can help. And for this, I'll kind of walk you through a story that we have here with Mrs. Hogan, who died suddenly and unexpectedly. He was an athlete, and I share these pictures and the story with the family's permission. And actually, they did a whole Wired article on this family. And so if you are interested, you can track that down. It's on the internet somewhere. But Hogan died suddenly while playing sports, and he had 3 siblings, Ethan, Kayla, and Austin. All of which had no idea they had disease. Ethan and Austin also have the same disease Hogan has, which is arrhythmic cardiomyopathy, and have ICDs in and have had significant arrhythmias, and Kayla has been fine. And so how is it that these individuals have disease? And by the way, if you give them the arrhythmic cardiomyopathy gene test, they're negative. And so we really didn't know why they were sick. And so we've been studying them using these induced pluripotent cardiac myocytes. And so with a few drops of their blood, we've made their cardiac myocytes. And this is that example I showed you earlier. This is what those cells look like. Sorry. Let's see. So I can get it to play for you all. They beat in a dish, just like cardiac myocytes do. And this provides a really unique model from which we can learn about disease and test new therapies. And just to kind of share some early data that we have about this. So these here to the left are cardiac myocytes, and we can actually treat them to induce fat inside those cardiac myocytes, just like patients with arrhythmic cardiomyopathy have. And these pink blobs are basically fat molecules that build up within these cardiac myocytes. And you can see the mutant line has a lot more cardiac myocyte fat than the wild type line. In addition, we know that one of the reasons that the arrhythmias happen is that calcium leak occurs, very similar to CPVT. And you might see kind of these white dots here, these kind of white flashes in these line scans, all of which shows calcium leak that's not present in individuals who are healthy. And we have early evidence, and I'll share some of it here, that we can actually provide a drug that's targeted toward their mechanism of disease to actually suppress the calcium sparks that might lead to their arrhythmias, and some early evidence that we can actually suppress the fat that deposits in those cardiac myocytes based on this new therapy. So we're super excited that if by studying this family and learning more about their disease, we might be on the path to new small molecules, new medicines that potentially might have benefit down the road. Again, a long ways to go, but I think highlights some of the potential of scientific advancements, particularly in the area of induced pluripotent stem cells, modeling individuals' disease. I'll also note that gene therapy trials are ongoing. There's one that you may know about through the SADS Foundation that's enrolling. And so if you have arrhythmic cardiomyopathy or interested in learning more about potentially gene therapy trial, specifically for patients with PKP2 mutations, which is the most common mutation in AC, check out the SADS website. Very exciting stuff. What about BRUGADA syndrome? How can we treat BRUGADA syndrome? It's really challenging, right? No good medications to prevent the arrhythmias. Unlike long QT syndrome, where there's non-specific beta blockers, you really can't use those for BRUGADA syndrome. And so, can we develop new therapies for BRUGADA syndrome? And this is where I think gene therapy actually has an incredible role. So gene therapy, as Dr. Etheridge already mentioned, is using genetic modification of cells to produce a therapeutic effect. And I think when I think of gene therapy, I tend to think of, oh, what if I swap out a disease-causing mutation with a normal piece of DNA? And that's one approach, but there are many other approaches if we're smart about how we do this. That may not involve changing the genome at all. Well, what if you could deliver a missing gene or a missing protein into a cell? We talked about BRUGADA syndrome being a loss of function of the SCN5A-encoded sodium channel, and half of the sodium channel is simply not enough. And so what if we put the sodium channel back in? We wouldn't have to change the genome, and maybe we could take care of this disease. Well, some of our best strategies to do that are now available, and they involve things like a virus called an adenovirus. That adenovirus is really good at finding heart cells and really good at injecting messages into those heart cells. And in theory, could inject a message that encodes a sodium channel, encodes a gene. However, to the left is a picture of the cardiac sodium channel, Nav1.5, the sodium channel that's lost in BRUGADA syndrome. And it is massive. It is a huge protein, way too big to fit inside a little tiny virus. And so this was not our idea. This was an idea of a very talented colleague of mine, Anand Bursak here at Duke. It's like, well, if we're limited by size, why don't we think small? Why don't we find a sodium channel that's smaller than the cardiac sodium channel? And if we can show that we can find that ion channel, and if we can show that it works, maybe we'll have a potential therapy down the road for BRUGADA syndrome. And so not just think small, he actually thought very small and looked in bacteria. And what Nanad found was this. Down here at the bottom is what we call BACNAV. It's a bacterial sodium channel. That's its structure. Up top is the cardiac sodium channel, the one in your heart and mine, and it's really big, but if you kind of break down the shape of it a little bit, it's actually 4 repeating subunits, one of which is found in bacteria. So we thought, well, maybe what if we could assemble this small little channel, put it into a virus, show that the virus can infect safely heart cells, and then express that ion channel. And if it actually makes it and can conduct sodium, we're in business. Because then basically we could take somebody who has an SCN5A mutation or a loss of function and is only making half of their normal sodium channels and replace that other half with BACNAV. All right, that's the idea. And share some very exciting results. These are still very early. These are cells and these are in tissue and in isolated things, but I think some very promising results. And so this is results from our paper. These are cardiac myocytes to the left, individual heart cells, right? And all the ones that are green have BACNAV. And so we're able to take a virus and infect these cardiac myocytes with BACNAV. That BACNAV is made and glows green in the cells. We're then able to see that the BACNAV gets to the membrane, the outside, which is exactly where we want to go. And we're able to show that that BACNAV can make a sodium current, can actually take over the physiological effects of SCN5A. So down here to the left, these downward swoops is sodium movement inside a cell. And the panel all the way to the left is without BACNAV. The panel all the way to the right is with BACNAV. And the amount of sodium it lets in is comparable to a regular heart sodium channel. And so again, we are nowhere near giving this to patients, but I think the concept of using a bacterial sodium channel to maybe treat a human disease is at least possible. I'm very excited about this. And again, this is work that Nanad has been leading. And finally, you've heard a little bit about Mike Ackerman's long QT syndrome approach from Dr. Etheridge. And that's basically to prevent these arrhythmias using gene therapy. And again, here too, you're kind of stuck with the genetics of the disease. So you can't just re-express the potassium channel in the setting of long QT type 1, because you still have the mutant sodium channel kind of gumming up the works a little bit. And so what Mike's team has done, and this is kind of a picture of what his team has shown, is that if long QT syndrome type 1, you've got this mutation in KCNQ1. This mutation actually causes that bad protein to be expressed. And so you can't just overexpress normal protein because you still got the bad one, right? And so what he has come up with is a strategy for actually silencing, shutting down that bad copy, and then turning on the wild type copy. And that's something he's termed suppress and replace, which I think is a pretty clever way of doing things. And this is evidence that this works. So these are action potential tracings of individual heart cells. And these action potentials are akin to an ECG, but for a cell. And just as long QT has a longer QT interval, cells that are, quote, from long QT patients will have a longer action potential. And here, since your x-axis is time, what you'll see is a longer action potential means that that line goes out further on that x-axis. And you can see the red line here, for example, is that long QT, that prolonged action potential. You can see it kind of lasting longer in all of these tracings. But then when they suppress and they replace, that normalizes, goes back to what looks to me to be pretty close to normal, which is really exciting. And they quantify it here to show that they're doing a good job of shrinking that QT interval in the individual cells. So super exciting work. As Dr. Etheridge was mentioning, it's going to require a number of years. It has to be tested in animals first, and then safety has to be shown in humans, but at least in cells, very exciting evidence that perhaps this might be a way of the future. And so I hope I've shared with you maybe where we are at the beginning of a very long road. I'm excited. I think from a scientific perspective, even in failure lies opportunity. So if all of these advances that we're talking about don't make their way directly to you in the next 5 or 10 years, I know that we'll learn a lot from those failures and those mistakes, and that'll allow us more opportunities down the road. And if any of these are able to help any of you in the next few years, that just, I think, is the reason that we all do this. So my take-home points, this is an exciting time for science. new advances in gene sequencing, genome editing, gene therapy, and creation of patient-specific models, I think will lead to improved diagnostic and improved therapies. And discovery is a team sport, and we need you on the team. This is our team here at Duke. Some of these people, I've highlighted their work, and these are our funders off here to the right. I'd like to thank you for the chance to be with you today. Welcome any questions.
Thank you so much, Andrew. And we do have 8 questions posted in our Whova platform. So I'll start with those and we'll just take them one by one. So where would you recommend a computer engineer with experience in algorithm development and AI go to support SADS research?
My goodness. Well, I'll try not to be too excited. We always love to collaborate. Drop me an email. We'll talk more. But I think you're at a very important interface. So I think the idea behind using computational sciences, artificial intelligence in a multidisciplinary way to help patients, we showed some evidence on the genetic side, but also on the diagnostic imaging side. Any application like that, there's a lot of interest, lots of excitement. And I think finding your way to those academic centers and being able to find teams that are willing to think broadly about how we can communicate and collaborate, I think is the key. But it sounds like you're in a perfect spot.
Thank you, Andrew. What is the best way to get involved in clinical trials if your EP does not participate in that space?
There are a number of different ways. So if your EP doesn't, you can simply ask him or her who might be a good person to reach out to. Many foundations, like you saw one example where SADS actually is a conduit for plugging in patients. So I'd talk to SADS, peruse the website. We talked about the ARRHYTHMIC cardiomyopathy gene therapy trial. And also it's something too, you can take a look online and see. I know Duke oftentimes highlights clinical trials that their physicians are part of so that we can kind of break down the barriers to access to care that way. And so I think doing a little bit of discovery work on your own, talking with the SADS Foundation and your provider can be very helpful.
Okay, thank you. From Melanie, wondering what research we might see for non-symptomatic, non-presenting children with detected PKP2 gene.
So you mean children who might have the mutation, but so far have no symptoms and show no signs of disease, I presume?
Yes.
So that's
feels like a ticking time bomb.
Yes, no, I know. And the challenge there is we know that gene mutation is a risk factor. And we know that exercise, lots of exercise over a longer period of time can actually, worsen disease. And so you've got these children who are sort of sitting there with this mutation, no evidence of disease, but we're really worried that, how they progress. This is where I think gene therapy really has a unique role. I think we are obviously early in that sort of nascent effort to figure out new gene therapy techniques. But I think for those individuals, there potentially could be a gene therapy role. Now for children, and this goes back to some of Dr. Etheridge's earlier comments, for children who show no signs of disease, for those who show no symptoms of disease, honestly, they will probably be the last ones that we want to give a new gene therapy trial to, mainly because they're small. There's issues regarding children as a vulnerable population we have to keep in mind. Plus, they're not showing evidence of disease. It'll probably start with the most advanced patients where therapies are really limited. But my hope, my dream someday is that we can identify patients like this early, treat that disease before it ever has time to manifest. That's many, many, many years down the road, but that's my dream.
I know there are a few drugs for CPVT in the pipeline. When would you expect them to become available?
So CPVT specifically, because of that calcium leak from that big channel sitting inside the cell, RYR2, there are a lot of efforts to try to block that channel with different medications. I think those medications fall into 2 big categories. One of them are medications that are kind of already around that can be repurposed to treat this disease. Those medications typically take less time to get approval, but are still probably a few years out. Then there are the brand new medications that we would not have thought of or have to be invented or have to be sort of brought through the FDA pipeline. Those are probably further out. I'd say probably a few years for the earliest ones.
Okay, thank you. Are you familiar with the GPD1L as it relates to BRUGADA.
Yes.
It was tagged as a variant of unknown significance.
So GPD1L, you're absolutely right, is one of those BRUGADA syndrome genes. There's been a lot of debate about GPD1L. Some people say that we have really good evidence that some patients might have rare causes of BRUGADA syndrome from that particular gene mutation. There are others that say, well, they're very, very rare, and what we see are a lot more of are variants of uncertain significance in GPD1L. And I think probably for every one patient with a real BRUGADA syndrome GPD1L mutation, it's probably many, many more with VUSs. And that's, I think, the case with a lot of rarer SADS genes. Really hard, right, to say what's just there and what actually might cause disease. When we took an AI-based approach, we went right for the common genes because honestly, it's a little bit easier. But I think if we're going to get really smart about this, we have to know all the genes, including the rare ones. To my knowledge, there is no AI-based effort for GPD1L, but maybe if that person who asked me that first question drops me an email, we can take a look at GPD1L.
Okay. For VUS testing, can you use IPCM research to determine whether or not it is a VUS? On a related note, can wait a minute, just lost it there, it moved. I lost the rest of that question. Sorry, let me.
That's okay. The first part was a really good one.
Okay.
So the answer is yes, but with a caveat. So those cell lines can be used, and there are many investigators who say, hey, look, if we don't know whether a mutation causes disease, could we use CRISPR, make it just that one mutation into a normal line and look for evidence of disease? Or if we think we have a VUS in a cell line already, CRISPR out, change out to a wild type to see if it goes away. And if we can do that, then we can show conclusively the variant does or does not cause that disease process. Very common approach for investigators to take. The trouble is it's expensive and takes a lot of time. So in theory, it's possible to show that experimentally. Whether that translates to every person with that variant is an unknown question. And it can take many years and it's quite frankly really expensive. So that's why I think approaches like AI might be something to think about. But iPSCs absolutely, to your credit, have a role there.
Okay, on a related note, here's the rest of the question. Can IPSC-CMS be superior to gene testing? This came from Anita.
From gene testing, well, so I think the role for those cardiac myocytes that we derive from patients are those where there's some unanswered question. So for example, the family that I shared who had that sudden cardiac arrest, their unanswered question was, what's the cause of our arrhythmic cardiomyopathy? And so in that situation, yes, the iPSC, they had a gene negative evaluation. And right there, the clinical gene testing ends. We have no idea why they're sick. But if we can use their cardiac myocytes as a tool, those cardiac myocytes carry everything from that patient that's in their DNA. And that allows us the opportunity to try to figure out, well, what actually might be the cause? And what's the cause of their disease? So that's where I think the roles of cardiac myocytes are, in figuring out ones where the gene test is negative. Or to the question that came before, figuring out potentially where the variants of uncertain significance are something to worry about or not.
Okay, thank you. You referenced this in your talk. At the last conference, a physician discussed future genetic testing, altering the genes in rabbits with long QT to preempt human studies. Has this happened yet?
I think that would be a great question for Dr. Ackerman. I know he's very enthusiastic about that. He's the expert there.
Okay. Is there free software available to generate protein folding structure associated with my individual VUS in SCNF5A? Can you share the name of such software if it is available? And this came from Rachel.
Rachel, that's a great question. So as we showed in one of the slides I had, there are lots of sort of free things online. They have fancy words like SIFT, PolyFan, PhD-SNP, things like that. And they're there to kind of predict on the academic research side whether mutations might indeed be causative. These are not very good. The accuracy for these is well below what you would want to make a clinical decision about your care. We can use them sometimes with a lot of other data to come up with a kind of a guess, but they're quite limited. Even the data I showed you with AI, we're closer, but I don't think we're quite there. We're going to be wrong 10 to 15% of the time, even with artificial intelligence. And so, at least right now. And so for me, I think I'm going to want more than just AI or more than just a model. I'm going to want a deep phenotyping, so seeing a specialist, ensuring you have the same disease that people are saying that you have, using that clinical evaluation in your family history to view the genetic test differently, and perhaps there's AI there to help. But I still think that approach is the best way forward.
Okay, thank you. What are the updates on gene therapy?
Yeah, so we talked about a lot of them. It's basically trying to different ways of countering the genetic defect of those cells. I think globally, if you look at what's been done, there's a lot of work done in cells. There's some work done in small animals. There's even less work done in larger animals. And right now you're just seeing the first trials for humans. And those are really sort of just in their what we call nascent phase. They're just starting out. So that's kind of where we are globally with gene therapy. We're probably many years away from having that be ready for prime time.
Thank you. Last question. Do you see many JUP mutations in ACM patients in the US? In Germany, I was told that they are all VUSs and nobody knows if they are disease causing. Class 4/5 mutations from Ruth, who's in Germany.
Ruth, that's very interesting. I hadn't heard that. It's great to have people to share international perspectives. I think JUP is a little bit like GPD1L. It's one of those rare genes associated with arrhythmic cardiomyopathy, which makes it really challenging, right? Because if you have a rare genetic cause, but patients with variants in those genes are more common, just because we all have differences in our genome that make us individuals, figuring out what's disease positive is really hard. So I think for those kinds of genes, those rare disease genes like JUP, you're more likely to get VUSs, and to get that one that you know is disease causative, much harder to find, much harder to be sure of.
Okay. Well, that concludes the questions that I see in our Whova platform. Thank you very much, Andrew, for being a part of us and helping out our families understand the research updates.
Pleasure to be here. Thank you.
All right. Take care.